تفضل يا دكتور.
Good evening, everybody. Today is the last day in the annual meeting organized by Saudi Society for Clinical Chemistry. And in the first workshop in today's meetings and sessions is going to be the virtual booth industry workshop. We have 3 speakers today, and I'm so pleased to introduce them. Our first speaker is Dr. Munir Raggas. And Dr. Munir has received his PhD in biomedical sciences from Wright State University, Ohio. Dr. Munir also was an assistant professor of pathophysiology at Qatar University. Before that, he was also assistant professor at the College of Pharmacy, Al Ain University, and he was a research assistant in the Department of Neuroscience, Cell Biology, and Physiology at Wright State University, Ohio. Dr. Munir is currently the EMEA Scientific Manager at Beckman Coulter, leading and executing strategic scientific educational programs. He is also a member in multiple societies, the American Heart Association, the American Society for Microbiology, Society for Neuroscience, and the American Physiological Society. Dr. Munir has published cutting-edge research in the area of ischemic stroke treatments and has been granted the Original Work Award and Best Presentation Award for his research. Today he will be presenting about the Beckman Coulter Total Solutions to fight against COVID-19 pandemic. And with that, I ask that you give Dr. Munir your full attention, and I will invite Dr. Munir to start his presentation. Dr. Munir.
Thank you, Dr. Hassan. Thank you so much for this nice introduction. Assalamu alaikum, everyone. Hello, and thank you for the opportunity to be with you today. It's a great pleasure, basically. My name is Munir, and I'm leading scientific marketing activity, as Dr. Hassan mentioned. And today I'm really excited to spend with you the next 20 minutes, about 20 minutes, to quickly share with you Beckman Coulter solution to fight against COVID-19 pandemic. We are strongly committed in Beckman Coulter to this battle. Our highest priority here is to find solution and to ensure that we bring the highest quality tests to serve the need of our patients and communities. So to start the agenda here, I will go over 3 main topics, like very quickly. The first objective will be to understand the pathophysiology of SARS-CoV-2 infection. And secondly, we will discuss the current and future role of molecular and serological testing in overcoming this pandemic and how governments are now beginning to make use of these tests to open society. And finally, we will review Beckman Coulter solutions in the fight against COVID-19. So just to set the stage, COVID-19 has changed life as we know it. As we are speaking now, this pandemic has touched over 64 million people around the world, and this virus has claimed about more than 1.5 million lives across the globe. One challenge with this pandemic is that it is not easy to determine who has been exposed to the virus and who has not. Because, as you know, the virus sometimes can cause symptoms and sometimes will not. So old people, basically, and other people with underlying conditions like cancer and diabetes, they usually are likely to progress to severe illness. So if we look at the majority of COVID-19 cases here, we can see that over 80% infected people are asymptomatic, or sometimes they show just mild symptoms. And we can see 14% they will show severe symptoms, and 5% will end up in the ICU. This is very challenging, basically, when it comes to infection tracing and infection control, because you cannot tell who has been infected. So to understand how IVD testing is used to diagnose and to monitor COVID-19 infection, it's really important to understand the pathophysiological cascade of the infection. So if we look at this diagram, we look at the mechanism of viral infection in general, we can see that physiological phase starting basically with a viral hijacking the cells. And then also we can see another phase, which is the pathophysiological response, which basically a dysregulated immune response. So in the early stages, as you can see here on the left side of the screen, the virus will invade the cells, and the RNA basically will be inserted inside. For this process to happen, the virus need to use its receptor binding domain, or the RBD, of the spike protein to connect with receptor cells on the alveoli cells, which is the AC2. And then after that will be replicated and released, so can affect other cells. This attack will cause, definitely here, if you look at the left side, will cause something called cytokine storm, which basically releasing a lot of different cytokines like interleukin-6 and TNF and INF and others. That will be produced in high volume and leading to sepsis and leading to multi-organ dysfunction and eventually death. So if we want to categorize the lab tests that used based on the purpose in monitoring or diagnosis of COVID-19, we can see here in diagnostic tests, we have basically molecular tests and we have antigen tests at the early stages. And also we have for the management, we can use the serology testing, which is basically IgG and IgM. And finally, for monitoring patients, we could use some biochemical tests and hematological biomarkers, basically to indicate the progress of the infection. So, as you can see here, different tests, different formats are or have been used in different places, and they have their advantage and disadvantages. I just want to basically here highlight the importance of the medical value of the SARS-CoV-2 spike protein. It's really important we understand that there are different types of antigen that expressed by the virus, like spike protein and nucleocapsid and others. But it looks like some assays they are using in their design spike protein, and other they are using nucleocapsid. So I just want to mention here that Beckman Coulter, they are decide to go with the spike protein because we understand from multiple studies the medical value of this specific domain, and many studies already have shown that it plays the most important role in viral attachment, fusion, and entry to the cells, and so it can serve as a target for development of therapy and also for vaccines. Here I'm sharing with you two independent studies that looked at correlation between the antigen-antibody binding activities. And as you can see here on the right side of the screen, this study showed that RBD-specific IgG antibody response are rapidly induced in infected patients during the first 6 days after PCR confirmation. So these responses result in neutralizing antibody that directly correlated with the RBD-specific IgG titers. This is really important and very promising for ongoing studies, especially for a study on the seroprevalence in population. On the other side, in the left side, you can see another study that looked at the RBD-specific antibody titer compared to surrogate of neutralization potency in acutely infected patient. And again, it's just confirmed that basically there is a real correlation between the titer of antibody and this RBD. So to go further here, I just want to mention another thing about how spike protein is the ideal target for vaccines. So basically, based on this fact, we decided to go with that, and also with vaccine in development now, they are, or the majority of them, they are using this spike protein, and specifically this RBD domain as an ideal target for vaccine development. Obviously, because there is this kind of antigen basically will induce some antibody that will interfere with viral binding to the cell and thereby will neutralizing the virus. So on the next slide here, I want to show also the value of using a separate or standalone IgG and IgM assay in comparison to the total antibody assays. So obviously here, as you can see, our assay, it's a standalone assay. And the reason behind that, because when you use a total antibody assay, you cannot distinguish, or the clinician cannot distinguish between the early IgM and the late IgG, which kind of like tricky. So they have to go back and reflect to BCR or standalone IgG test, which is, as you understand, has many negative impact on that workflow. Here we are looking at some benefit of serology testing. So there are many benefits of using serology testing. Basically, antibody detection with PCR. This will complement the test and will provide a complete clinical picture for the clinician. It's also important to know who has been exposed and also will have a high value for therapeutic uses, especially for making the convalescent plasma, as well as for seroprevalence, as I mentioned, and vaccine deployment. So a lot of benefit for using a serology test. Now I want to go with you and overview the Beckman-Coulter solution, different solutions that we have to fight against COVID-19 pandemic. Some of them already released and some we are developing right now. So if we look here, we have released already the FDA authorized assay for detecting IgG, which is a qualitative assay. This has an excellent sensitivity and specificity and great throughput and onboarding stability. The nice thing also about this assay, it will measure or will give you result in non-reactive, reactive, as well as in acute vocal. So this is very important in the sero conversion. The other or the second assay also that we released during this pandemic was the Access SARS-CoV IgM. Again, this is another qualitative assay with a great performance. And for this assay, we are using the spike protein, the same target. And also, again, it can complement the PCR testing and provide more confidence in clinical assessment at early stage of the disease. And just recently, we have developed a semi-quantitative assay, which is IgG assay, that also got FDA approval or authorization. This assay will give you, the clinician, additional tools to evaluate the level of immune response and the IgG antibodies, and to establish a baseline based on the numerical results. So this is a great one, especially when it comes to vaccine development. So in this assay, as you can see here, has a great positive agreement and has 99.9% of negative percent agreement. This is really, really important because it will extremely lower the false positive rate. So this is, again, with high throughput and with high sensitivity and specificity. Now, here just to give you an overview about what we can offer. So all these tests that I just mentioned now will allow for a seamless integration and high efficiency for the workflow of the lab. So it will minimize, first of all, the time, and basically because we use the random access mode, which is the meaning of that is you do not have to do any batching here. The second thing will be providing one of the highest throughput in the market. So up to 200 tests, and you can run up to 4,800 tests per day, depending on the analyzer that you're using. The other thing is the ability to review patient results from both diagnostic and serology tests when you use a Remisol advanced midware. This one will help, again, for more efficiency of the lab work. Now, in this slide, I want to discuss a little bit about the SARS-CoV-2 antigen tests in general. So you might hear about the public health now experts. They are pushing toward changing the thinking about this, COVID-19 testing. So basically, they encourage looking for a different way to test for this COVID. So basically, what the experts think that they're calling for this shift about the testing. They think it is better to get fast and frequent results from any test that are reasonably accurate, which is basically majority of antigen tests, than just waiting for more precise results to be delivered after days long delay. So here, just general information about the antigen test. It's basically a different test. It's completely different from the PCR. It will look for the viral protein instead of the RNA. It has many advantages here. If we can mention some. So basically, it will be easy to run. It doesn't need any complicity or training, basically. So majority of them are easy to run. They have fast turnaround time, and also the good thing about them is the lower cost to do when compared to the PCR. So far, antigen tests are authorized by FDA for only to be used on the symptomatic individual. So no antigen test has been yet authorized by the FDA for use on asymptomatic individual. Here are a few scenarios in which SARS-CoV-2 antigen tests may reasonably be used in addition to testing symptomatic patients. So basically here it can be helpful to rapidly triage a patient in the emergency department. So these are all recommendations from the CDC, but again, there is no claim or there is no approval or authorization from any FDA or other regulatory bodies. So here's the scenario. So any congregate setting, as CDC described them, which is basically prisons, long-term care facilities like nursing home, and also any long-term living facility and correctional facility or rehabilitation facilities could be a good place or could be a good setting to start doing this kind of screening test using this cheaper and fast time around test, which is the antigen test. Another application or reasonable application of this test will be for remote population and in the area where there is limited access to the PCR, or when the time for or turnaround time for the PCR result is prolonged. So it will be reasonable to use this antigen test. But again, we just have to remember that this kind of test, basically, especially if you use them with asymptomatic population without any confirmed case, probably will require to be repeated multiple times. So this way they will alleviate the lower sensitivity of antigen test. So in addition to that, I want also to mention another important thing that has been considered since beginning of this COVID-19, which basically many studies suggested that COVID-19 patients, especially the acute one and the severe cases, may experience cytokine storm syndrome, which basically lead to sepsis and finally cardiac events. So for that reasons, also it would be reasonable to measure inflammatory biomarker like interleukin-6, like procalcitonin and high-sensitivity troponin I. All of them are critical for the prognosis, and also Beckman Coulter, we offer all of them on the same axis family analyzers. So to summarize the last slide here, just to give you an idea about Beckman Coulter coming test and also the one that already released. Again, we are so committed to this fight, and we have developed and we are developing more critical tests that will be released soon to help with the management, with monitoring and surveillance, and as well as for vaccine deployment and also for therapeutic using different tests like IgG for pre-screening and IgG also quantitative for evaluating the vaccine effectiveness of efficiency, and also antigen tests can be used also at the patient level for discharging, as well as to support the decision or to support decision when combined with the PCR and IgM test. So that's what I wanted to share with you, and thank you so much for your listening.